All right, so we will talk about variant filtering. This is after we've called our variants with haplotype caller, and we now want to refine our call set to adjust for the sensitivity and precision that we want for our given task, given the data we had. So the philosophy of the variant calling algorithm is to be as sensitive as possible, to emit as many variants within reason, so that analysts, depending on their task, can adjust to a precision that makes sense for them. So if they're, only want really high quality variants, then they can take the sensitivity down and have a really precise call set, whereas if they want any signal, they can tune that however they like. So in general, there are three filtering approaches supported in the GATK. The simplest and most straightforward and always available is to just do a hard filter, which is where you have some numeric cutoff on an annotation value, some kind of summary statistic that describes each variant. The more sophisticated approach is to use something called the VQSR, which builds a Gaussian mixture model and tries to model what high-quality variants look like in an annotation space. We'll talk about in more detail. And then lastly, we have a convolutional neural net, which looks at the annotations, but also looks at surrounding sequence context as well as the raw reads in order to determine what's a real variant and what's an artifact. Both of the modeling approaches require truth data. So we need validated VCFs, which are not available for every species. But we now have high quality validated VCFs for humans. So we're able to train these models and deploy them with the GATK. So filtering is always going to be a trade-off between sensitivity and specificity. And so these different approaches may give you a better response, but a filter can only reduce the sensitivity. So the job of discovery is left to haplotype caller, and here we're just worrying about precision. So a little bit of history. In the beginning, we had only these hard thresholds when we realized that our call sets also included variants that were of dubious scientific nature. So we can think of these as handcrafted decision boundaries. And they often work very well. The problem is they tend to scale very poorly. So once you have a new data set, that magic number that you found was a good threshold on your previous data set for separating the good variants from the bad variants often doesn't translate as well. Furthermore, you notice in this picture, all the lines are straight. And that's because when you set a cutoff, you're creating like a vertical line where everything to one side you let through and everything to the other side you block. And we have many different annotation values that we can compute on these variants, but these hard thresholds don't interact in any way. They're just these blocks that divide up the annotation space. So that's a little bit blunt. It's not, you're not able to see interactions between the variants, between the annotations. The sort of next step in variant filtering history was handcrafted features or handcrafted annotations, these numerical statistical summaries of each variant, and then a machine-learned decision boundary on top of that. So we would compute some features. I'll say features and annotations. I'm using these words interchangeably. What I mean is some kind of number that describes the variant. Maybe it's just the depth of reads that supported it. Maybe it's some kind of measure of how biased the variant was. If there were if the variant always showed up in the beginning of reads or at the ends of reads, some kind of numeric description of the variant. And then based on a set of those numbers, we learned some kind of decision boundary to separate the good variants from the artifacts. Moving along, we decided maybe those annotation values don't really encapsulate everything there is to say about a variant, all the information there. So maybe we can, rather than from the top down deciding what are the interesting features or annotations to describe a variant, let the machine learning system determine for itself what makes a good variant. And so this is the machine learned features. And then after the machine learns those to compute those features, we again have a machine learned decision boundary to separate the good variants from the bad variants. And then the sort of final step that we can envision, which we have not yet reached, would be sort of machine learning the whole thing. So not just the features, but the actual architectures. And we'll get into more detail about what those architectures are really doing in just a minute. So here, another word about these annotations or features. Again, there's just a list of numbers that describe the variant. Some are totally self-explanatory, like AC, number of chromosomes carrying the alt allele, or DP, the depth of coverage. MQ is the root mean square of the mapping quality of all of the reads at that site. So, and there's a whole zoo of numbers like this that the GATK can compute for you. Two others that deserve mention, which aren't on this slide, is the strand odds ratio, which determines if the variant was seen more on one strand versus the other, and the read position rank sum, which determines if the variant was seen at a certain point in the reads. So it turns out that reads get lower and lower quality as they go along. As the sequencing experiment continues, the sequencer gets tired, or the chemicals in the sequencer get tired. And so the bases that you get at the end of a read are actually, in general, of lower quality. So if you're only seeing a variant at the end of a read, it's a little bit suspicious. And we can summarize that all in a number. Okay, just to look at the hard filtering picture in annotation space is, again, drawing these hard cutoffs where we have, here are all our variants, and then this is some annotation, here's another annotation, and we see them in this 2D space. But because we have a hard threshold, all we can do is just cut straight lines through here and exclude one of the quadrants or let one of the quadrants through. Compare that to a learned model where we can have a much more complicated decision boundary, where we can really look at interactions between the two different annotations and decide if a variant should stay or go based on all the evidence at once, not just considering each piece of evidence separately. So models are better than thresholds. Okay, but as I said, for those models, we're going to need truth data. We need a validated VCF that we can trust that tells us what is a good variant and what's a bad variant. So how do we do this? Well, the first approaches were people sitting in a room and looking at IGV and saying what they thought looked like a good variant. But pretty quickly it became clear that that wasn't going to scale. So the first approaches that really scaled were consensus-based. So we would run a bunch, or the government, NIST, would run a bunch of different variant calling algorithms. GATK, Freebase, a whole soup of different variant calling algorithms on the same sample, and then look at where all the calls agreed. And that would become a validated call here in green. Where some of the algorithms had calls and others didn't, we would just remove that region from the confident region. We would say, we're not sure what's going on there, and we would just exclude it. So this is a very good method at getting high-quality data. There's a very dangerous aspect to these kinds of methods in that you only focus on the things that are easy to call, the things where everybody is already doing well. So it sort of narrows your view of the genome. Platinum Genomes was a project by Illumina where they also used this consensus approach, but they additionally brought in a pedigree of a large family from Utah that had extensive pedigree information. So they had 3 generations sequenced, and they were able to look at not just consensus between different algorithms, but also the Mendelian violations. So they could assume certain inheritance patterns because they knew the pedigree, and when that was violated, they were able to remove those calls from their validated VCF. This more recent approach to generating truth data is really interesting. So we're going to quickly dive into this. This is a project that came out of the BroadE. And it begins with a interesting biological phenomena, something called a complete hydatidiform mole, which is it's a human egg that has no DNA, that gets fertilized by sperm, and the sperm's DNA duplicates and starts growing a tissue. It can be dangerous, so these are terminated, but the tissue is haploid because there was no DNA from the egg. So it's like it's a human genome, but it's haploid. So it's much easier to call because there are no heterozygous sites. So this tissue was sequenced, and the crucial part of this experiment was it was sequenced with long-read DNA sequencing technology, not with the Illumina short reads. So it's a totally orthogonal kind of technology, and because the tissue is haploid, it's much easier to call. The problem with long-read sequencing is it tends to have many more errors. But when it's a haploid organism, it's so much easier to call. It's much easier to filter out the errors. So from 2 of these different haploid organisms, or tissue, that got sequenced, we were able to synthetically create a diploid, where we knew what the true haploid sequence was of each. So this is a way of constructing heterozygous looking, like a diploid seeming genome, where you really know the truth data for each of the parental chromosomes. And it has no dependence on the Illumina technology. So it's a really interesting truth set that doesn't have those issues of only looking towards the consensus. And this is another one of the validated VCFs that we use to train our models. OK, so the first model available in the GATK for variant filtration is the VQSR. And this is based on Gaussian mixture models, where we have an annotation space, and we're showing 2 dimensions here. But it's important to keep in mind this space is as big as the number of annotations you wish to include. So typically this is like a 7 or 9 dimensional space where you have those read position, rank sum, the strand odds ratio, the depth, your favorite annotations to describe your variant. You throw them all in there, and then the Gaussian mixture models fit Gaussians to all of the variants that are recognized from the validated BCF. So all of the known truth sites get these nice Gaussian blobs fit onto them. And then you look at everything that's sort of left over, and you actually fit more Gaussians onto that. And then to determine if a new variant is real or an artifact, you look at which, does it fit in the nice Gaussian blobs that described your true data, or does it fit in the leftover Gaussian blob that described all the remains? So this is a great method. Because when I came, my first task was to design the successor to this. I've spent a lot of time thinking about its shortcomings. So it's a great method, and I'm not trying to insult it, but I am going to point out a few problems with this approach. So one is that the annotations are not, there's a big assumption here, which is that our annotations are going to fall into these Gaussian blobs. And sometimes that's true. But often the annotation values, for example, mapping quality. Mapping quality is just arbitrarily cut off at 60. So if you plot mapping quality values, you don't see a Gaussian blob. You see something with a very hard edge right at 60. So when you try to fit a Gaussian blob to that, you run into all sorts of problems. Similarly, the negative data, well, positive data in general tends to be, for many annotations, is well described by Gaussian. But the negative data is often just everything else, and it's much harder to describe that with a nice Gaussian blob. So, yes?
So, the data you're showing here, is this like the gold standard data that you're trying to fit to, or is this like an actual data set?
Yeah, so shown in red, this is an actual data set. Shown in red is everything from the actual data set that was also seen in a validated BCF. So most of your variation is common. So if you get sequenced, most of the genomic variants that we find are going to be things we've seen before in the general population.
The Gaussians are fit on the validated data set?
The Gaussians are fit on your data set, but they're only fit to the things on your data set that were also in the validated BCF. And that's what's shown in red here. What's shown in yellow is all the things in your VCF that were not in the validated VCF, and there we fit different Gaussians, which we keep separate, and then the final sort of score that this filter emits is the ratio between those two Gaussian mixture models.
How do you figure out how many Gaussians to fit? Because I see you are showing 4 there. Why not?
Indeed, yeah. So that's what we call a hyperparameter. And it's something you give to the model. We have default recommendations that work well. It's, I think, 7 by default for SNPs and 5 by default for indels. But sometimes, if you try to fit too many Gaussians, the tool will crash because it starts like fitting a Gaussian onto one point, and it becomes, the Gaussian explodes into infinity. So that can be tricky. Yeah, so it's a trade-off between how much expressivity you want in your model and how much data you have to support that. So for small call sets, you often have to take that number of maximum Gaussians down. Any other question? Yeah?
So probably for like exome data, let's say, you should put the Gaussian values lesser?
Yes, and typically you use a bunch of exomes when you train this model, because you want lots of data in order to see these patterns. Any other questions on VQSR? Okay, so another thing to notice about this model is we're just using the annotation values, okay? So we're leaving some data on the table. The first data item that we're leaving on the table is the DNA sequence. So the sequence context matters. And I'm going to illustrate that by showing a Lego plot. How many people have seen Lego plots before? Okay, great. So you know how to read it. If you haven't, it's just the colors indicate what the SNP or what the mutation was at a particular site. The boxes within each color indicate what the neighboring base was on each side of that pointwise mutation, okay? So this corner is an A to G SNP that was surrounded by 2 T's. Okay, and the important thing to notice is that this is not a flat landscape. We have a skyline in here. There are sequence contexts which lead to much more mutation than others. This is not at all uniform. So this type of information is not at all accessible to VQSR because the annotations are computed based on that location. And in particular, let's look at this tower. This is the A to T SNP where you're surrounded by 2 T's. Okay, so it's like you're in a poly T or a poly A part of the genome. And those homopolymers, where you have the same base over and over again, are notoriously difficult to call and notoriously difficult for the sequencer to properly sequence. So there's many errors in contexts like that. Okay, so we would like to bring this information of the DNA sequence context into our model. But how do we do that? We have to somehow find a numeric representation for the DNA sequence context. Well, luckily, there is this one-hot tensor encoding, and it's a very straightforward encoding where we take a sequence of DNA and we replace it with a matrix with 4 rows and as many columns as there are positions in the genome that we want to look at. And we call these different columns the channels. So we have a channel for A, C, G, and T. And the last thing to point out is we can encode ambiguity as well. So if we have like an N, which could be any base, that's just quarters down. Or if it could be a G or a T, but it's not an A or a C, we can encode that in this representation. Okay, and now we want to operate on that thing. We've now figured out how to encode the reference. How do we do some operations on that? We'll motivate that with this position weight matrix, where we look at a multiple sequence alignment. And we notice that the sort of conservation is distributed across the sequence. There are parts of the sequence that are much more conserved than other parts. Okay, one way to represent this multiple sequence alignment is just by consensus, where you take the majority at each position. But tons of information gets thrown away if you do that. It's a tyranny of the majority, because you can't see that this G and T are much more important for this motif than the surrounding bases. So rather than just having 1s and 0s, we can replace these with floating point values that encode our expectation of seeing the base at that site. Now, with this position weight matrix, we can go looking for sequences. So this operation is called convolution. We have our sequence here, and then we have our motif here. And by multiplying this one by this one, and then shifting it over, we're doing something called convolution. And that's the same as detecting motifs. This is motif detection. But it's a clearly defined mathematical operation. Now, people have known about this for a long time, and the first way we got position weight matrices was by observing them, by finding those multiple sequence alignments and observing what sites were more conserved than others. Since we now have this truth data, we're able to learn the motifs dynamically for the task at hand, and our task is filtering variance. When we do that, this very reassuring thing happens, which is we come out with the motifs that we've already known were important. So for example, we said how the homopolymers are very difficult to call. Well, one of the very common motifs that comes out when we train these 1D models on the reference sequence is these homopolymer detectors. So the model's learning to look at the sequence context and see, oh, is it all a bunch of C's? In that case, I'm pretty likely to have some errors. We also see more complicated sequence context detectors like short tandem repeats. And this is like what I call a thymine edge detector, because you have positive T and negative T. So if there's a poly T section that ends, this motif is gonna be detected. Okay, and let's see, does that? We're learning many of these in parallel.
So you're doing these on the validated data?
Yes.
So and you're just automatically trying to find
So it's a
frequency motifs?
yes, and again, it's we look at a new data set compared to the validated data set. So all of the things that overlap are true. All of the things that are not in the validated VCF are false. So we have true and false, and we train it to distinguish the two. And it's learning many motifs at once, and you see here's another poly A, here's a CT, 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 a short tandem repeat detector, a poly G. So lots, another STR detector here. But we're learning many of these at once. So the model learns how to find all kinds of different sequence contexts and put them together for this task of variant filtration. Okay, so that brought in the, yeah?
Just in the motifs, what's with the negative T? It's like, it's less probability of having a T in that position. What's the negative and positive?
Yeah, so negative is, in a way, the sign is hard to interpret because we're looking at an early layer of a model, and if later on there's a different sign, it will switch. So sort of more significant than whether it's upside down or is the size. Yeah, exactly. But eventually, at the very end of the model, the negatives you can think of as saying it's more likely to be an artifact, and positives are more likely to be a true variant. But because we have this distributed model, deeper layers can switch the sign, so it's not a straightforward interpretation.
Another thing, going back to your Lego plot, how do you know that these are not actual more frequent mutations, but more frequently artifacts? Like, how do you know that this signal is not real?
Yeah, we don't. I mean, we don't know from this plot, and the model will have to learn, because it's true that some of these processes are just true over evolution. So we wouldn't expect a uniform Lego plot if we looked at perfectly pure data. The hope is that that's encoded in the training data. So the model will learn to distinguish what is the expected distribution on real variance, and what is something that looks like that we only see in the artifacts.
And basically, this is what you're doing to teach the motifs, right?
This is more just motivating that sequence context matters. Like, why did we think it was important to give the model sequence information? It's because we'd seen these Lego plots and we knew. Okay, great. So that gave us sequence context. There's a whole boatload of other data left on the table, which is the raw reads themselves. So these models that we're showing, these are the 1D model, which you will play with in the workshop in an hour or two. Next, we have the 2D model, which is more powerful but slower. And what does the 2D model do? The 2D model also brings in the raw read data. So previously, we only had the sequence context. Now we add everything, or not quite everything, but all of the reads that came directly off the sequencer. So here's an IGV screenshot of a heterozygous SNP. Here you see in red the T's, and here the reference was a C. And there's a lot of other information in IGV that's available. So the different color of the reads shows their strand. You can see where on the reads, and each base in each read has a quality that the sequencer emitted. So we take all that information, and similarly to how we did for the reference sequence, we encode it in this thing we call the read tensor. So now it's not a 2D matrix anymore, it's a 3D tensor. But similarly, it's encoding the sequence information, but now sequence information from the reference and from all the reads, as well as the base qualities and some additional information that comes with the reads, like their mapping quality, if they were optical duplicates, reverse strand, if they were first in pair, second in pair. And here we're seeing the different channels. So before our channels were just A, C, G, and T. Now we have all these different channels for the read metadata as well as the sequence context. Okay, and now let's just look at some read tensors. Here's another SNP. You see it's a G to A SNP. So it's right there is the SNP. Here's the brown is G. This is A. It's heterozygous, right? So we have the reads separating nicely, half and half, diploid organism. And then to the right and to the left is the surrounding sequence context, which we know is sometimes informative. Here are some insertions and deletions. Here we have a heterozygous deletion. So you see all this is missing from our sample, but it was in the reference. So it's a deletion with respect to the reference, but only on half of the reads. So it's a nice heterozygous deletion. Here we have a homozygous deletion. You see all of the reads are missing. That purple is not one of, is just to show we don't have one of the 4 bases. And then here we have a heterozygous insertion. You see the reference now is purple because there's no reference sequence there, but one of the parents had extra bases. And here is a homozygous insertion of a T, where we have all of the sample had this extra T, but the reference didn't. Okay, and now, as we were sort of alluding to with the discussion of positive and negative, we're going to build many layers on top of these first motif detectors. So we look at motifs in the raw data, but then we look at how those motifs work together. It's a little bit easier to interpret models like this that were trained on images because we're so used to looking at images and understanding images and not so used to deconstructing read tensors. So let's look at the images first. You notice in the early layers of images, we extract edge detectors. These are very basic, primitive features important for doing all kinds of visual tasks. It's to know where there are edges, what are the contours. Then we start looking at patterns, very regular structured patterns. Then you sort of get into these textures. And finally, you start getting into object parts and then full objects. So similarly, as we build up a hierarchy on these read tensors, and here we see a lot of poly A's, and here you see something that's detecting heterozygous versus homozygous reads. Okay, so we put it all together into this model. We have, again, our read tensor with all the reads, all the sequence context going through these layers of convolution. we bring in our old friends, the annotations over here, and at the end, we say, is it a real variant or is it an artifact based on this validated truth data? And we compare this to VQSR as well as to other approaches, and this is the area under the precision recall curve. So this is a way of analyzing where we don't have to pick a threshold. We look at any given threshold, how is our sensitivity and precision, and we're doing quite well. We look at how this generalizes across models, across data sets. So this is not just cross validations, it's cross generalization. We're looking at exomes versus genomes, HD38 versus HD19, different sequencers, and over here we see this sort of generalization across all these different data modalities. Okay, the tool itself is actually broken into two tools. There is a CNN score variance, which just adds a field with the score, and then a filter variance, which allows the user to set a specific threshold and filter the variance based on it. Any questions?
So all the layers you use in your deep network are convolution layers, or do you?
Great question. Yeah. So this whole part is convolutional, and it's getting small. The representation is getting smaller and smaller and smaller. So here we have like 128 by 128 goes in. But by the time you get here, it's like 16 by 16. So we've got this more global view. And then we just flatten it. So it just becomes a vector. And onto that vector, we concatenate the raw annotation values, which are also just a vector of numbers. And then the rest of the model is something called a multi-layer perceptron. It's just dense layers. And this is trained jointly over SNPs and indels. Rather than VQSR, you train a separate model for SNPs and another one for indels. Because we want to use all the data and benefit from all the data at once, we train a model across both types of variant.
And 128 is the context that you provide?
Yes.
Have you tried larger and smaller?
Yeah. So 128 seems to be the best for this type of model. When you also bring in the read pairs, so you get a little bit more global information, 256 performs a little better. It's much more expensive computationally. So 128 is like the trade-off we're working with for now. But in general, if the bigger, the better. But there starts becoming a lot of extraneous information that you're bringing in too once you're looking really far downstream or upstream. So it becomes harder to train as well. There's also this 128 is a sort of arbitrary cutoff of the maximum read depth. So if there are more than 128 reads at a site, we randomly downsample. Yeah?
You said there is a second part of the tool, which is filter variance. And so we give a threshold. But at the beginning we said we don't want to give a threshold. So what's the
Yes. So we don't want to give a threshold on a particular annotation value. But eventually, you have to make a call, right? So the way we've structured the tool is this CNN score variance takes all the information, evaluates the model and all the different annotations, looks at everything, and then summarizes it in a single number that's the score. And it's added to the info field of the VCF for each variant, and it'll say CNN1D equals 1.2, or CNN1D equals -5 if it thinks it's a bad variant. And then, so that number encapsulates everything from what the model has learned. And it's on that number that you eventually, yes, you do have to make a call. At some point, you need the threshold, but we want to push that decision as far back and leave it up to the analysts. So if they're just hoping to get any variant, then maybe they'll take that threshold way down. So, and specifically, the threshold is in these SNP tranche and indel tranche arguments. And that's specified in terms of desired sensitivity on a truth set. So 99.9 means I want to get 99.9% of the known variants to safely come on board. So as you take that number down, your false positives go down, but your false negatives go up. Yeah.
Like best practices thresholds for these?
Indeed, yeah. Yeah, the tool comes with reasonable defaults for human data, yeah.
Has it been tried on non-human data?
It has. I mean, on diploid organisms, it's reasonable to do. We're not sure what you'll get if you try it on tetraploid things.
I have a diploid organism, so would you recommend trying this as opposed to one of the other approaches you mentioned?
Sure, if it was a single sample run, it definitely worth trying, yeah.
But not from any sample run?
Yeah, so if it was joint call, if the annotations are joint called, then we still recommend VQSR, because these models were all trained on annotations from single sample runs. Thank you.